All right, in this video, we're going to begin talking about mutations. We're actually going to spend the next 3 videos talking about mutations because there's just a lot of content here and there's a lot of different kinds of things that can go on. So remember, normal function of the cell is to have the DNA transcribed into messenger RNA, and that messenger RNA be translated by the ribosome to make a polypeptide. That polypeptide will then fold up into a particular kind of protein, which has a special kind of shape for a special kind of function. If all goes well, that is exactly what's going to happen. And so gene expression then creates a particular phenotype in that organism, whether it's blue eyes or height or whatever. All right. Any number of things that make sense and some that are more abstract. Changes in that genetic code are called a mutation. All right. So mutation has a negative connotation to it just by the sound of it. It sounds like it's going to be a bad thing, but all mutations are not bad. Some of them can be just neutral. Some of them can be beneficial. Actually, this is the main driving force behind evolutionary change is the mutation of the DNA. Without the mutation of the DNA, there is no change. And so now if those changes are good, then that organism will do well. If the changes are not, it won't. We'll talk more about that in a later video. So big idea here, you can see here. So here's a normal cell, makes normal sorts of proteins, and they do normal sorts of things. But in a cell that is, say, a cancer cell, well, you have some mutations. Well, those mutations are going to lead to differently coded RNA, which is going to lead to differently shaped proteins, which is going to lead to different functions. And so in this cancer cell, perhaps what's going on here is these are MHC1 and 2 are proteins that allow cells to interact with one another from outside. If those cells are no longer able to interact with one another, there's some sort of function that is going to fall off, and that's not going to be a good thing. Particularly just if cells need to know, okay, I can stop growing now because there's cells near me. If they keep growing, well, that's what causes a tumor. And so moving on. So this idea of being a negative or a beneficial mutation is important because the mutation is not a purposeful thing. Mutations happen accidentally. They're not, in a perfect world, the system that we've talked about would work flawlessly and there'd be no problems. However, you have 6 billion base pairs, and the expectation that they would all be transcribed and translated 100% perfectly is just a bit high. And so mutations are going to happen, and they're considered neutral if the protein doesn't change. That can happen. We're going to see an example of that in a minute, where the mutation occurs, but there's no real change in the protein, so there's no real change in the phenotype and the organism overall. Example of this is, not an example of this. We'll talk about the beneficial one. So here's an example. This is called a pepper moth, and this moth used to live on these white trees, right? And so this white moth was very well camouflaged. And then the industrial age happened, and these trees were covered with soot. This is a real life example. And there was a mutation. Well, these particular moths are now going to stick out like a sore thumb, and anything that eats moths is going to come and have field day, unless there's a mutation that causes the coloration to change. Again, the moth didn't go and hatch a plan in order to be better camouflaged. This is an accidental mutation, but it just so happens that this mutation is very good for the new environment that has been presented. Well, consequently, what happens is that natural selection is, industrial age ends. They realize that putting soot in the air is probably not the best. They clean the trees up, and now the black moths stick out like a sore thumb. And so this has been an interesting thing for evolutionary biologists to watch, but it all has to do with this presence of this mutated gene or not. We're going to look at some different types of mutations. Essentially, any sort of thing can happen. And so, again, a mutation is a change on the DNA. This shows a particular string of DNA, which codes for a particular string of RNA. This codon, remember, 3-base unit called a codon. We use the codon chart to decide that lysine is the particular amino acid that needs to be present. Here are some different mutations. They're called mutations because it's a change in the DNA. Instead of a C, I now have a T, right? Which causes me to have 3 A's, which, that still codes for lysine. So this is a mutation that's a neutral. It doesn't cause a problem. Sometimes you'll see it called a silent mutation. All right, in this particular instance, though, a mutation causes an early stop codon, which would be bad. There would just be no protein produced or half a protein. All right. And in this particular, both of these over here, I'm not sure the difference between a conservative or non-conservative. Probably has to do with the chemistry of the amino acid itself, which is not important to me or this class. But you can see what's going on here. Rather than get lysine, we get a different amino acid altogether. Particularly if you're supposed to have a basic amino acid and instead you have this polar amino acid, it's going to cause a shape change and it's going to cause some significant problems with that protein. If you change the shape of the protein, you change the function of the protein. You get the idea. Another type of mutation is where a base can be inserted or deleted from. So it'd be like if I told you to read the first 50 pages of a book and someone came and took out 30 pages out of the middle. Well, you would read the 20 pages you were supposed to, and then 30 more pages that you weren't supposed to read, right? You would get way ahead of yourself. But because you were just reading 50 pages, you didn't know that something had been taken out. And so if something gets taken out or added to the DNA is still going to be read in these groups of threes, and it's going to make particular amino acid sequences. Here's is the same amino acid altogether. But notice they just put a single A in here, right? And instead of cat, you have, I'm not going to even try to pronounce that, but you get the idea. Something changes, and this, you'll see this called a frameshift mutation sometimes. But a frameshift mutation can be particularly bad because everything after it or before it can be wrecked. Here's an example of something that a mutation can cause. It's called sickle cell anemia. Sickle cell anemia is a disease that affects the shape and the function of red blood cells. Red blood cells carry oxygen, which is obviously a very important function for us. And if a single or a single DNA sequence, so here instead of a T, you have an A, which means that you're going to have a valine instead of a glutamic acid, it's going to cause such a problem, a single base, that your cells aren't nice and plump and able to carry oxygen. There are these odd sickle-shaped cells that get stuck in your blood vessels, which, if your blood vessels are clogged up, that's not good for anybody. And so sickle cell anemia is a particularly bad disorder because it affects the amount of oxygen that you're able to get to your blood. And so this is an example of how that single change can affect an organism phenotypically.